What is a cell made up of? We learned earlier that all cells have smaller structures inside them to fulfill specific life functions. These structures are called organelles. But even under the most basic microscopes, most cells display three components: the nucleus, the cell membrane, and the cytoplasm. The cytoplasm is a clear ground jelly-like substance that fills the space inside a cell. Now let us look at all of these one by one. The cell membrane. As mentioned before, every cell has a structure that preserves its structural shape and integrity. The structure encloses the cell and is called a cell membrane. The membrane also fulfills other functions. It allows for the selective transport of nutrients and waste materials into and out of the cell respectively. How does this transport happen? The cell membrane is selectively permeable, which means that only certain substances can travel across it. Its structure allows for the transfer of certain molecules but not others. Certain chemicals like carbon dioxide and oxygen are transported via simple diffusion. Diffusion is a process where a substance migrates on its own from a highly concentrated place to a place of lower concentration. For example, a bottle of perfume, if broken inside a room, spreads the smell into the entire room. The concentration of carbon dioxide inside the cell is usually greater than the outside, and conversely, the concentration of oxygen is usually higher outside than it is inside. Thus, both of these gases move in and out of the cell on their own. Water also obeys the law of diffusion. When kept separated by a semi-permeable membrane, a selective membrane, water can migrate from a dilute solution to a stronger one by a process called osmosis. The larger solute molecules do not move across the membrane. Based on this understanding, we can now classify solutions as isotonic, hypertonic, and hypotonic. You may have noticed that if you take dried raisins and put them in water, they swell up. You may have also seen that if you take fresh fruit and add it to a concentrated salt solution, they shrink down. The reason for this is that the concentration of salts in that solution is greater, equal, or lesser than the concentration of salts and solutes inside the cell. If the concentration of salts or other solutes outside the cell is greater, the solution is said to be hypertonic. In such a case, water will migrate from inside the cell to the outside, causing the cell to shrink down. If the concentration of salts and solutes inside and outside the cell are equal, the solution is said to be isotonic in nature. Also, if the solution has a lower concentration of solutes and salts compared to the water present inside the cell, the solution is said to be hypotonic. In this case, the cell will absorb water from the outside, as you saw with the raisins. The cell wall. In plant cells, the cell membrane is further enclosed by another structure called a cell wall. The cell wall is mainly composed of cellulose in plant cells and chitin in fungal cells. The cell wall provides rigidity to the cell. If a plant cell is placed inside a hypertonic solution, the cell will start losing water, as we discussed earlier, and the shrinkage of the cells caused by this is observed and known as plasmolysis. The nucleus. The nucleus is the Latin word meaning the core or center of something. Fittingly, the nucleus is also the name of a cell component usually found at or near the center of a cell. The nucleus can be stained by a specific chemical dye to be observable under a microscope. The nucleus is a roughly spherical structure surrounded by a porous membrane called a nuclear membrane. The membrane contains a large amount of DNA, that is deoxyribose nucleic acid, which is composed of extremely long chains of repeating units of nucleic acids. These chains are further folded up again and again to form a very tightly wound up bundle of a substance named chromatin. The nucleus plays a central role in the reproductive process of the cell and contains all the information required to produce the cell. It should be remembered that the nucleus is a relatively late development in the evolution of unicellular life. Even today, there are large groupings of microorganisms that do not have a differentiated structure to hold DNA. In such organisms like bacteria, the DNA material is suspended in a loose bundle inside the cytoplasm of the cell. Such organisms usually also lack most other cell organelles. This group of cells is known as the prokaryotic group of cells. A more modern and developed type of cell is one where there is a nucleus and a larger variety of cell organelles. These are called eukaryotic cells. The cytoplasm. Under a microscope, it is observed that there is a region within the cell that does not take up a stain. This colorless or transparent region is composed of a clear jelly-like substance which we call the cytoplasm. The cytoplasm encloses the cell organelles in eukaryotic cells and encloses the cell membrane also. All eukaryotic cells have an array of highly specialized organelles, and they function very much like organs in large multicellular organisms, which perform specific functions. This kind of specialization is absent from prokaryotic cells. Let's look at some of these organelles one by one. The endoplasmic reticulum, or ER. The endoplasmic reticulum is a highly folded, highly intricate structure that looks like a large interconnected network of chambers and bags, which are also technically called vesicles. The ER membrane has a structure very similar to the plasma membrane and plays the same role. In this case, that of being the transport of various large and complex biomolecules. The ER is divided into two types based on the detailed structure of the surface: the smooth and the rough endoplasmic reticulum. The roughness of the RER, or rough endoplasmic reticulum, comes from small structures on its surface called ribosomes. These structures specialize in the production and transport of complex proteins. The smooth endoplasmic reticulum, or SER, is concerned with the production of fat and lipid molecules that are required for various biological functions and for the production and upkeep of cell membranes, which is called membrane biogenesis. The intricate folded networked arrangement maximizes the surface area over which the synthesis can take place, and the transport of these molecules can be effected. The Golgi apparatus. It consists of a system of flattened sacs, technically called vesicles, arranged in vertically stacked structures called cisterns. These vesicles often have connections to the endoplasmic reticulum and can be considered a part of the membranous network found inside a cell. The apparatus is concerned with the synthesis of biomolecules and for the transport of these molecules inside and outside the cell. For example, the Golgi apparatus can form complex sugars out of simple ones, and is also involved in the formation of another organelle called the lysosome. So, what is a lysosome? Lysosomes are structures which contain powerful enzymes synthesized in the endoplasmic reticulum. They are used for engulfing and digesting food particles, a process that requires enzymes and is called lysis, hence the name lysosome. If accidentally ruptured or broken, they can cause the cell to be digested and destroyed from the inside, which is why they also have another name. They are called the suicide bags of the cell. Mitochondria. Mitochondria are highly specialized and semi-independent organelles which generate a chemical called adenosine triphosphate. Adenosine triphosphate is the energy currency of the cell and the human body and other organisms as well. These structures generate this chemical, and this chemical is then transported to other points inside the cell. The mitochondria contains its own DNA and can synthesize proteins on its own. Their origins are still mysterious. We believe that these organelles are actually the remnants of bacteria that were engulfed by larger cells long ago and entered into a mutually beneficial relationship. Plastids. Plastids are in a way the plant counterpart of mitochondria. They are found only in plant cells, but they perform similar functions. There are two types of plastids. the chromoplasts, which include the colored plastids, and the leucoplasts, which are colorless or white plastids. The chromoplasts, if they are green in color, are called chloroplasts, which are the structures responsible for photosynthesis inside a plant cell. Leucoplasts have a storage function. Their purpose is to store complex sugars, lipids, and fats inside them. They can also contain their own DNA and have a limited independence. We come to the vacuoles at the end. Vacuoles are simply large sacs that contain nutrients or waste materials or foreign particles engulfed by the cell. They are present in both plant and animal cells. Vacuoles found in plant cells contain cell sap, which is full of sugars and other water-soluble nutrients. They are also much larger than vacuoles found in plant cells. Often they can take up to 50 to 70% of the internal volume of the cell. Certain specialized cells in animals like fat cells are also extremely large. Storage vacuoles can occupy up to 90% of the cell's volume. In plant cells, aside from storage, vacuoles will also offer turgidity and rigidity to the cell.